Okay, what are the silent features of, let's see the silent features of the human genome. The silent features of the human genome are that the human genome contains 3164.7 million nucleotide bases. Okay, and then every gene consists of 3000 bases, but sizes varies greatly, with the largest non-human gene being, that is the dystrophin. Okay, in average gene there are 3000 bases and the largest non-human gene जो है वो क्या है, dystrophin है. Okay, और इसके अंदर कितने 2.4 million bases present है. Okay, and it mean we can say that the total number of gene in human genome are estimated at 30,000. Total are 30,000. Okay, and almost all 99.9% of the nucleotide bases are exactly same in every human individuals. Okay, 99.9 तो same होते हैं. Okay, 99.9%. Okay, you can say 99.9% nucleotide are exactly same in all the peoples. Okay, and the function are unknown for over 50% discovered gene. Okay, and less than 2% of the genome codes for the protein. Okay, and the repeated sequence make up a very large portion of the human genome. Get it? Okay, and the chromosome 1, जो chromosome 1 है, chromosome 1, you can say that the chromosome 1 has the maximum number of gene, and what are the number of 2968, and chromosome Y has the least number of gene, that are 231. Okay, तो chromosome 1 had the maximum, chromosome 1 में maximum है. Okay, number of genes that are 2968, Y has the fewest 231. Okay, get it? And there are about 1.4 million location in human genome were single base DNA difference occur in SNP, single nucleotide polymorphism. And this information is helpful in finding chromosomal location for disease associated sequence and tracing human history. Okay, get it? And what are the applications? You can say applications for future challenge, and application that its knowledge is helpful in research involving biological system including human biology. इसका जो knowledge होगा helpful होगा research करने में biological system और human biology में. And with the whole genome sequence and newer technologies, we can be systematic in our approach to various medical questions on a broader scale. Okay, and all the genes in a genome, like example, all the transcripts in a particular tissue या organ या tumor can be studied. Okay, so these are the application in future challenges also. Get it? Now let's see the next, DNA fingerprints. Okay, DNA fingerprints, what is it? DNA fingerprint, you know that it is a very quick way to analyze the difference in the DNA sequence. Okay, the DNA fingerprint is a technique to find out variation in individuals of a population at DNA level, and it was first developed by the Dr. Al Jeffrey. Okay, and Al Jeffrey and works on the principle of polymorphism. कौन से principle पे work करेगा? Polymorphism in DNA sequence. Okay, so a DNA polymorphism is, what is it? It is any difference in the nucleotide sequence between individuals. Okay, and the difference can be single base pair changes, deletion भी होता है, insertion भी हो सकता है, और even changes in the number of copy of DNA sequences. Okay, so it works on the principle of polymorphism. DNA fingerprints involve identifying difference in a specific short nucleotide repeats called variable number tandem repeats. Okay, VNTR, that vary in number from person to person and are inherited. जो inherited होते हैं और इनका number जो होता है वो person to person क्या करेगा, vary करता है. And the VTNR of two persons may be of same length and sequence at certain sites but vary at others. Okay, get it? Vary होंगे. अब now how the DNA, okay, firstly DNA का क्या है, DNA का जो उसको isolate किया जाता है, extract किया जाता है, what is the process? First is the extraction and isolation. DNA is extracted and isolated from the cells in a high speed refrigerated centrifugation के through. Then amplification होता है. Amplification mean many copies of extracted DNA are made, okay, by the पीसीआर पॉलीमरेज चेन रिएक्शन के थ्रू बहुत सारे क्या बनाया जाएगा, उनको कॉपी करवाया जाएगा, डीएनए को एक्सट्रैक्ट करके उसकी कॉपीज होगी पीसीआर के थ्रू. देन डीएनए को कट किया जाएगा फ्रेगमेंट्स में. डीएनए कट कौन से वाले एंजाइम्स के कौन से एंजाइम से होता है, रेस्ट्रिक्शन एंडो न्यूक्लियोस एंजाइम. Then after separation of DNA sequence, तो cut DNA fragment are introduced and pass through the electrophoresis. सेटअप होता है इलेक्ट्रोफोरेसिस, ओके, जो कंटेन करता है एग्रोस पॉलीमर जेल. ओके, तो 10 जो सेपरेट फ्रेगमेंट को विजुलाइज किया जाता है, स्टैंड किया जाता है विद द डाई, ओके, विच शो द फ्लोरोसेंस अंडर अल्ट्रावायलेट रेडिएशन. देन सडन ब्लॉटिंग, द सेपरेट डीएनए सीक्वेंस आर ट्रांसफर्ड इनटू nitrocellulose or nylon membrane by placing over the gel. Then hybridization, the nylon membrane is immersed in a bath radioactive probes. These probes target a specific nucleotide sequence that is complementary to them. Okay, then autoradiography, the nylon membrane is placed to an X-ray film and dark bands develop at the probe site which is resemble the barcode. Okay, so this is the method by which DNA is extracted. Okay, get it? See the application of DNA fingerprints. Okay, technology, DNA fingerprint technology. It is used to identify criminals and rapists. Okay, and it is to solve the parental dispute, to solve the immigrant problems, to identify dead bodies of soldiers died in wars, to identify dead bodies of person died at accident and bomb blast, to identify racial group, to rewrite biological evolution, to detect inheritable disorders, and to detect donor cell in a case of transplantation. Okay, so these are the application of DNA fingerprints.